then you need to remember that. Okay. Now, most genes in your body, right? You have we are eukaryotes, so we have exons and introns, right? So you have exons, we have introns, and then you have your transcription, right? So you start transcription here and here. You put poly-A tail here, or not yet. You have your primary transcript or pre-mRNA, same thing, primary transcript. So you do transcription and you have your primary transcript. You're going to do RNA splicing, right? So you have your 5' cap, you have poly-A tail here, and then all the intron sequences are gone, and you have your mRNA, right? And then you're going to translate that. You have your protein. This one folds into whatever shape that you want it to fold, right? So that's what happens with most genes in your body. B cell receptors and T cell receptors do something that's different, which is that before you get to this part, there's another step that has to happen, and that's gene recombination or gene rearrangement. We use the two terms interchangeably, okay? So some of the exons that you're going to see here for the B cell receptor actually does not exist as a contiguous sequence. They exist as gene segments, okay? And somebody has to cut and paste, literally paste your gene, your DNA, to form a contiguous exon sequence, okay? This is different than RNA splicing because we are not cutting and pasting RNA. We are not doing the splicing in those terms. So when we say splicing, we mean RNA splicing, right? This is actually gene recombination. You're actually changing and messing your chromosome sequence. You're actually cutting and pasting DNA in your nucleus on your chromosome, doing gene recombination, and then you end up with a different DNA sequence, okay? Only B and T cells do this, okay? No other cell in the body does this, okay? So the way you're going to come up with 10 to the 11th different specificity is when you are picking the gene segments to cut and paste, you have many choices. And depending on which one you choose, you're going to end up with a different combination and therefore different exon sequence and therefore different protein sequence, okay? So the analogy that people usually get is, so say that you want to come up with 100 different outfits, okay? You can either go with 10 pairs of shorts and 10 different shirts, and then 10 times 10, you end up with 100 different outfits, or you could make the fashion choice to go and buy 100 different jumpers, okay? You still end up with 100 different combination or 100 different outfits, but it's probably a lot cheaper to just buy 20 pieces rather than buying 100 different jumpers, right? So you can work with fewer number of gene segments. You can mix and match and come up with lots of different possibilities. So that's what we do. We are doing this and not that system. So the antigen binding portion, okay? So the variable IG domain of heavy chain and variable IG domain of the light chain will do gene recombination, okay? So heavy chain does this and light chain does this. So you're going to have 2 different gene segments or 3 different gene segments, depending on which chain. You're going to cut and paste and produce that, okay? So for heavy chain, you actually have 3 different gene segments that are going to recombine or rearrange to give you the variable IG domain, okay? The gene segments are called V and D and J. V for variability, D for diversity, J for joining, although no one really remembers what they were supposed to be, okay? For light chain, you have V segments and J segments, okay? So heavy chain actually has 3 different segments, B, D, and J segments that have to cut and join together. Light chain has V and J, okay? 2 gene segments. Any questions so far? So when you're making a heavy chain, okay, you have, so the example I'm showing you like 4 different V segments, 2 different D segments, and 3 different J segment possibilities. Okay, there's way more, but I couldn't really spend time making like 50 different squares. So here they are. So say that you pick that one and this one and that one, and you put them together. Okay, that's your heavy chain. So when that is now transcribed, spliced, and translated, you end up with this heavy chain IG domain. Okay, so heavy chain IG domain. Light chain, you pick that one and that one, it comes together, you have that light chain. Okay, so you have your light chain. Whatever protein surface is formed by that is what is going to be binding to whatever antigen that is complementary to that particular heavy and light chain surface. Okay, now a different one. It's exactly the same gene segments. If you look at the color and all that, exactly the same, but you're picking something else. You pick this, this, and that, right? And you pick this and that. So V and J, that's different, right? So when this one is now made into a protein, you have this. When that one is made into a protein, you have that. So now you have a surface that's formed, and you're going to bind to an antigen that can form complementarity area to that particular heavy and light chain antigen binding. So putting the two together, you start with the same gene segments, right? So gene configuration is not changed, but depending on which one you pick, right? Which one you pick, you end up with this versus that. This one is going to bind to an antigen that's different than that particular antibody. So with the same number of gene segments and gene segment configuration, you came up with 2 different antigen binding specificity, okay? Now you can do this with lots of different gene segments. So immunoglobulin gene loci, so multiples of locus, have multiple V, D, and J segments, okay? So chromosome 22 and chromosome 2 has a light chain gene locus, and then heavy chain is on chromosome 14, okay? Light chain genes, there are actually 2 different chromosome locations that can encode the light chain. One is called lambda light chain locus, the other one is called kappa. You have 2 different parts that can make antibodies, or the light chain, okay? So it's 144. You have, you know what, we'll stop here because this one is a bit involving because I have like 1 minute left. So we'll stop here and we'll pick it up on Thursday. Thursday, we don't start with a quiz, we just go straight into the lecture.